So we'll provide a brief outline of our webinar to all of you, and I will quickly switch to Everardo. So indeed, Everardo will give you first an overview of phase 1 trials, and will give you some clinical insight into these trials. I will then try to look at benefits and limitations of various designs from a statistical point of view, and especially we will try to look at the efficiency question, which of these designs might be most suitable for most efficient design. And hopefully we will end up with some conclusions that might guide you with respect to the plans of your phase 1 trials. So without further ado, I will give the floor to Everardo.
Thank you, Tomasz, and hello, everyone. I hope you can hear me well, too. My task today is to provide you with an overview of phase 1 trials. And to the extent possible, I will try to make specific references to the various types of designs, because this will be covered by Tomasz. Although I have to say that most of the overview I will present refers to the so-called classic 3+3 design, just because this is the most commonly found design in the literature. So I'll try to give you some essential definitions, the main objectives of phase 1 trials, some of the prerequisites. I'll briefly discuss the main ethical aspects of phase 1 trials, discuss the most commonly used endpoints, a few words on the peculiar issues with targeted agents, and then a few considerations for special populations. I would like to recommend to all of you this book that appears in the reference to this slide. This is the book by Drs. Eisenhower, Twelves, and Mark Boyce from IDDI. It's a very practical book, and a lot of information that will be presented here can be found in that book. Sorry, just taking some time to pass the slides. So, So phase 1 trials sit at the entrance of the clinical phase of drug development. And you may have heard that for the past few years there have been attempts to conduct the so-called phase 0 trials, which aim mostly at assessing the pharmacodynamics of agents. And the main goal of these trials is to perhaps decrease the chances of failure further down in drug development. But we won't discuss phase 0 trials today. So I'd like to provide some preliminary definitions. And the first one is, what is a phase 1 trial? Although this sometimes will come labeled as such, for practical purposes, a phase 1 trial is a trial of a new agent or combination whose tolerability in man is still unknown or still not completely understood. And this means that sometimes a phase 1 trial may be the so-called first-in-man or first-in-human trial for a given agent or combination, but sometimes this is a second or third or even fourth phase 1 trial on the same agent and combination for various reasons. Sometimes phase 1 trials refer to let's say old agents that are now being combined for the first time, or even combinations involving at least one new agent. Sometimes they involve radiotherapy. This may need phase 1 trials at times, and at other times special populations may require testing of a new agent or combination, and I'll say a few words about that. On the other hand, we won't cover the so-called phase 1/2 trials, which are studies in settings for which there is previous experience to suggest that the phase 1 will be successful in other words, less concerned with toxicity, and at the same time, when it's thought that a direct transition to phase 1 to phase 2 is acceptable or likely to occur. So these are specialty trials that won't be discussed today. There is an essential tension that characterizes phase 1 trials, and this is indicated by these two opposing forces, so to speak. There is a major concern with toxicity in these trials just because the agent or combination is new. And at the same time, patients either implicitly or explicitly expect some therapeutic benefit. Sometimes not only patients, physicians themselves expect that benefit. So this is something that is always present, and we have to deal with this and manage this as best we can, because this will always be present when a phase 1 trial is contemplated or conducted. Individuals enrolled in phase 1 trials, unlike other areas in medicine, are typically patients, in this case cancer patients. In other areas in medicine, as you know, healthy volunteers are often the subjects included in phase 1 trials. Although I need to say that there are phase 1 trials in healthy volunteers in oncology, but these are usually trials with targeted agents, and more often these trials involve a single dose of the agent. So the typical patient in a phase 1 cancer trial has an advanced tumor for which no standard therapy is available. But these patients are the ones with good performance status, otherwise they shouldn't be included in these trials. The mechanics of phase 1 trials are such that these are typically single-arm studies, although in some situations it might be interesting to consider a randomized design in some settings. Just almost by definition, these are open-label trials, and more often there is inter-patient dose escalation. Although intra-patient dose escalation might be a good idea in some settings, and this has been done in the past in various situations, as long as it's not done to guide decisions. But it's something to consider in the attempt to offer active therapy, at least to some patients participating in these trials. So most patients end up receiving subtherapeutic doses just because the starting dose is quite low, and I guess Tomasz will say a few words about that when he discusses the 3+3 design. The ideal setting for a phase 1 study would be a specialized unit, but this doesn't need to be the case in all studies. So phase 1 trials may be conducted in the common oncology settings as long as people are dedicated to this. Often these are single-center studies, but again, there is no reason why they couldn't be multi-center studies as long as good communication is available. Many times companies decide to conduct parallel phase 1 trials, for example, one in North America and the other in Europe or Japan or other parts of the world, and this is common too. So practically speaking, there are three main objectives in phase 1 trials. The first is to characterize the safety/toxicity profile of a new agent or combination. A second very pragmatic goal is to determine a dose and schedule for further testing, for example, in phase 2 trials. And then, of course, it's important to characterize the pharmacological properties of this new agent or combination in humans, because this may not have been done before. So what are the prerequisites before conducting a phase 1 trial? There must have been extensive preclinical development that allows for human testing. And this refers to various aspects described here. Chemistry, structure, mechanism of action must be well understood. Ideally, there must be evidence of in vitro and in vivo activity. There must be extensive animal toxicology studies to characterize the agent or combination, and ideally the dose activity and dose toxicity relationships need to be well known. Other fundamental considerations include the starting dose, the schedule of administration, and who the participating institutions will be. As I said, there is an ethical dilemma in phase 1 trials essentially because the risk to benefit ratio is problematic, and various reviews over the years have attempted to come up with average expectations we should have when conducting phase 1 trials. The historical average toxic death rate is in the range of 0.5%, although there is good news in the sense that this has been improving in recent years. On the other hand, the average response rates in phase 1 trials are in the range of 5 to 10%. And however, there's significant variation across study types, but most patients, in other words, will not derive an immediate benefit from participating in such trials. On the other hand, patients and sometimes physicians expect some benefit, and some authors have tried to understand if this mismatch between reality and expectations could come from the phrasing of consent forms. And for example, in this study, it was found that more than 80% of patients expected benefit, and some even expected a cure. And the analysis of consent forms showed that it's not something in the consent forms themselves, because a direct promise of benefit is never made, or so cure is rarely mentioned. So there must be something else in human nature that accounts for the misunderstanding that exists in phase 1 trial participants. So the typical endpoints for phase 1 trials are the maximum tolerated dose, or MTD, the recommended phase 2 dose, and then the pharmacological aspects of pharmacokinetics and pharmacodynamics. So let's take a look at each of them in turn. First, let me provide you with a definition of dose-limiting toxicity, or DLT. So in phase 1 trials, DLT is often defined as an adverse event that is deemed unacceptable and hence limits further dose escalation. Of course, this adverse event has to be at least presumably treatment related. So most of the times the DLTs are defined as grade 4 or greater hematologic toxicity, or grade 3 or greater non-hematologic toxicity. Although you should have in mind that these definitions are often used, but they may be changed if there's reason to suppose that a trial, for example, may have neuropathy as a significant or likely adverse event, and then other grades of this event may be considered, just because it may be irreversible in some cases. So the definition of DLT may be protocol specific. And also, we should have in mind that DLT often refers to what happens during the first cycle, and hence phase 1 trials are not a very good setting for assessment of more chronic toxicity. There is, of course, a relationship between the MTD and the recommended dose for phase 2 trials, and this is mostly historical. What's important is to have these definitions in place before the trial is begun. But historically, in the United States, the dose limiting, the MTD has been defined as the dose level not causing dose limiting toxicity in more than 1/3 of patients, and hence for these trials, the recommended dose for phase 2 testing is the MTD itself. Whereas in Europe and in Japan, historically, the MTD has been defined as the dose level causing DLT in more than 1/3 of patients, and therefore the recommended dose for phase 2 trial is one dose level below the MTD. In terms of pharmacokinetics, I thoroughly recommend you to take a look at the chapter by Dr. Christwells in the book I mentioned. He's an expert in the clinical pharmacology of cancer drugs, and the points he says we should consider include whether a single dose versus repeated dosing is being tested, whether this is a trial in cancer patients versus healthy volunteers, whether special populations are being considered, whether pharmacokinetic interactions need to be investigated either with other drugs or with food, and also issues related to the analysis, what kind of material or specimen will be used. Is it blood, plasma, urine? How is this going to be collected and processed, and what assays will be used? Likewise, for pharmacodynamics, some points to consider include, first, a question: what is the role of pharmacodynamic assessment in your trial? Is it to provide proof of concept that the agent hits the target? Is it to guide dose escalation or to define recommended phase 2 dose? These two are very good ideas, seldom used in practice, but very good in theory at least. Is there a search for a biomarker? This is often done later in phase in drug development, especially predictive biomarkers, but it could be a good idea to start searching for biomarkers early on. And finally, is it to establish PK-PD relationships, which might be very relevant down the line in terms of refining the dose of the agent or combination. Also, what is the method of pharmacodynamic assessment? Is it tissue biopsy, whether a tumor or normal tissue? Is it blood or plasma? And whether functional imaging may be used to help in this assessment. So this is a good time to discuss potential differences between so-called classic cytotoxic chemotherapy versus targeted agents. Let's first take a look at the left-hand panel labeled as A here, which depicts the typical dose-response curves for cytotoxics, both in terms of the dose activity curve in blue and dose toxicity curve in red. So for typical cytotoxic agents, there's a steep relationship between in these two, there's a steep behavior in these two curves, and there's often a separation between them in the sense that at a given dose, one is likely to find activity at the same time as the toxicity is still not too high or preferably low. So this is the scenario for a typical cytotoxic agent. Whereas for targeted agents, all the other three situations are, at least in theory, possible, and it's hard to tell in advance exactly what will happen unless there is extensive preclinical data in that regard. So for this panel here, we can see that there is a steep increase in the dose activity curve at the same time as the dose toxicity curve behaves in a different fashion, and this would be a very favorable scenario in the sense that the activity is achieved at the expense of relatively little toxicity. This may be the case for a drug for which the toxic effect may not be mediated by the same target as the activity or anti-tumor effect. However, these other two scenarios are possibilities. On the right-hand side, in the top panel, we see a situation where the agent will hit the target in a saturable way, such that both target saturation and anti-tumor effect have a shoulder in the curve, whereas toxicity may not have the same shoulder, and in this relatively unfavorable situation, the curves are really near, and toxicity might preclude anti-tumor activity. Whereas in the bottom panel, we see a more favorable situation in the sense that at a time when the target is close to saturation or anti-tumor effect is close to the shoulder, there is relatively little toxicity. So all these scenarios are possibilities, and phase 1 trials will help in understanding which is the case for particular agents or combinations. So other issues with targeted agents are what kind of dose-response relationships are being encountered, and how will this be used in terms of designing and analyzing the study. So essentially, dose escalation with targeted agents could be guided by pharmacodynamics. However, this is very seldom done. In a survey of 60 completed phase 1 trials, most of them used the regular or the typical parameters used in cytotoxic chemotherapy, namely toxicity. Sometimes pharmacokinetics was used, but very, very seldom were pharmacodynamics or functional imaging used to determine the recommended phase 2 dose. And this would be something to consider, because this might make the trial more attractive in many regards. Another essential consideration in targeted agents is whether patients should be selected for the presence of the target. We often see phase 1 trials for which no selection was made, and it might be good to consider early on whether the population should be enriched for patients expressing the target, as long as this is well characterized. And finally, I'd like to say only a few words about special populations, because these may require separate phase 1 studies. And these populations are children and adolescents, and patients with either liver or kidney dysfunction. So just 2 points I wanted to make. The first is for pediatric phase 1 trials. There is a very nice correlation, as you can see here in this graph, between the MTD in children and the MTD in adults. And this allows for starting doses that are higher in children. Since phase 1 trials in children are usually conducted a few years after the adult corresponding phase 1 trials, this may be capitalized on, and studies may start with higher doses, and therefore fewer children will receive subtherapeutic doses. And the other consideration I wanted to make is about trials in patients with liver dysfunction. I just wanted to mention that there is a specific NCI classification system for hepatic dysfunction, and this may allow for the stratification of patients enrolled in such trials. So now I'm going to hand it back to Tomasz, who will cover issues pertaining to trial design.
Okay, thank you, Eduardo. Yes, so I'll try to now focus a little bit on the design of phase 1 trials. There's a range of dose escalation designs which might be considered for the use in phase 1 trials. I've listed several of them. You can find more of them in this reference. However, I will mainly focus on 3 designs which are listed as first ones. So I will mainly look at the classical 3+3 because it's still very often used and perhaps abused. I will also take a look at the continual reassessment method, which is a method which has been established, which properties have been established for a long time, and it's a good compromise, although it has some practical disadvantages which may discourage, unfortunately discourage clinicians from using it. And then I will try to say something about new method, new design, which was proposed quite recently, which sort of like tries to combine the ease of implementation with good properties. So if we talk about the dose escalation designs, there has to be a dose escalation scheme, and traditionally the so-called Fibonacci or modified Fibonacci scheme was used? Well, in this design, the initial dose is used based on the animal data, for instance, like 1/10 of the lethal dose, 10 in mice. And then the doses are increased relative to this initial dose according to, as I said, the so-called modified Fibonacci series, which I will just show in a second. And then the maximum tolerated dose, as Everardo mentioned, is a dose for which a given proportion of patients, like 33%, experience dose-limiting toxicity, for instance. So this Fibonacci scheme, well, this is the so-called Fibonacci series, which is known in mathematics. What is perhaps most important is that this series is characterized by these relative increments. So we start from the initial, let's say, dose, then we increase it by 100%, then we would increase it by 50%. And as you can see, the next increase is stabilized by around 61%. Now, the modified Fibonacci, which is used in phase 1 trials, is modified in the sense that, as you can see, initial increase is 100%, but then the increases are smaller than as compared to the original Fibonacci series, and as you can see, they stabilize at 33%. So it means that with when going to the higher doses, essentially we increase the dose by 1/3 compared to the previous dose. So this is like a scheme of the doses. So we can propose in a phase 1 design to assign the doses according to this modified Fibonacci scheme. Now, in the classical 3+3 design, the doses are given to the patients who are assigned in cohorts of 3. So this is a design of this is the scheme of the classical 3+3 design. So we start at the lowest dose, then we treat 3 patients at this dose level, and we look at the number of those limiting toxicities among these 3 patients. If there is no toxicity, no DLTs, we just go to the next higher dose level. If we see more than one DLT, we say, okay, we've reached the MTD, and then we stop the trial and we make the decision regarding the MTD. Now, if we see 1 out of 3 DLTs, then we treat 3 more patients at this dose level. And then again, if in these 3 extra patients we see no DLTs, we go to the next higher dose level. On the other hand, if we see at least 1 extra toxicity in these 3 extra patients, then we say, okay, we've reached MTD, and then we stop the trial and modify, and we recommend appropriate dose for the phase 2 design. So as you can see, this is a very simple design. It's easy to implement, and essentially, from the practical point of view, there's not much problem with it. There is, however, several problems with it in terms of both ethics and statistics. Now, from the ethical point of view, it appears that there are too many patients who are treated at low or subtherapeutic doses, and this is like an exemplification of the tension which Everardo mentioned, that you would like to put, well, the patients or clinicians expect some benefit from the treatment in phase 1, and if you start to if you give patients subtherapeutic dose, the benefit is unlikely. Also, dose escalation may proceed too slowly, especially if you start with a too low initial dose. I mean, it may take a lot of time to reach the level of MTD, and it means that you may need to put too many patients on too low doses, and this will result in a too long trial and maybe too costly trial. From a statistical point of view, there are several issues. First, there's actually a large uncertainty about recommended dose because too few patients are treated near or at recommended dose. Essentially, you can have as few as 3 patients treated at such a dose. There's no formal justification for this ad hoc design. Essentially, there's nothing related to the optimal dose selection or efficiency which is actually underlying this design. And also, there's no guarantee that the MTD corresponds to a given percentile of the dose-response curve because there's no underlying dose-response relationship for this design. So essentially, we cannot really say whether the dose which we selected corresponds to a particular percentile. So this is an example of a classical design. As you can see, there are like 13 doses which might be used in this design, and you see that it may take you a lot of time before you reach the level where some toxicities indicated by the squares are appearing. And this essentially means that you can only you are reaching the level where the toxicity may be appearing only by, for instance, in this case, like 25th patient. So it means that it may cost you 25 patients with the time required for treating and enrolling them until you reach the area where you may actually, where your MTD might be. So this is clearly one of the issues, important issues in terms of efficiency of the 3+3 design, which needs to be taken into account. For this reason, there were modifications of the classical design proposed, and I show here one of them, which is called 3+1+1 design, where we again start at the low dose level. You treat 3 patients at this low dose level, and then you look at the number of DLTs, and if there are none, then you essentially go to the next higher level, or you stop if you reach the maximum dose. On the other hand, if you saw 3 out of 3, then you say, okay, I stop because I identified my MTD. If you see 1 or 2 toxicities out of these 3 patients, you add 1 more patient. And again, if you see 1 out of 4 in total, then you go and increase, or if you are at the maximum dose, you stop. Or if you see 3 out of 4, then you say, okay, I identified MTD. On the other hand, if you have 2 out of 4, you treat again 1 more patient, and again, if you have 2 out of 5 DLTs, then you increase. If 3 out of 5, you stop and you identify your MTD. There are other modifications which aim at increasing the properties of the 3+3 design, and here I have perhaps a little bit too complex slide which tries to explain some operational characteristics of these different modifications. So here there are different designs. The red line is the classical 3+3 design. The gray line with dots is the 3+1+1 design. The idea rule is the black solid line, and essentially in this case we have like 7 dose levels, and our MTD is the 4 dose level, so the level at which we have 33% of toxicity, DLT. Now, these curves show the cumulative probability of assigning a particular dose or smaller doses in the trial. So the ideal rule, which is indicated by this black solid line, this ideal rule would put no patients at doses 1, 2, 3, and it would put all the patients at dose 4. And it means that it would put 100% patients, of course, at dose 4, and cumulative probability for doses 5, 6, and 7 will be also 100%. So this is the ideal rule. Now, if you look at the 3+3 rule, so the classical design, the operational characteristics are indicated by this red line. So we can see that the cumulative probability of putting patients at the dose 4, so at the MTD or lower doses, is about 67%, whereas the cumulative probability of putting patients at dose 3 or smaller is 33%. So essentially what this means is that only 1/3 of the patients are put on the MTD, and another 1/3 of the patients are put at the doses which are lower than MTD, and they may be subtherapeutic. On the other hand, you can see that there's also 1/3 of the probability of putting patients at the doses which are higher than MTD, so these are perhaps too toxic levels, right? If you look, on the other hand, on this 3+1+1 rule, so this is this curve, you can see that it has like about, it is 33% cumulative probability of putting patients at dose 4 or lower, with relatively low proportion of patients putting at doses 1, 2, 3. So you don't really put many patients on subtherapeutic doses. So, but again, it's about 1/3 of the patients who get assigned to the MTD. And this means that essentially, similarly to the 3+3 design, this MTD is selected only 1/3 of the cases. So this shows that essentially the modifications of the 3+3 designs are not very much different in terms of the selection of MTD. what you can see that here is in this legend you can see also the expected sample size for these rules. So what you can see, for instance, that the 3+1+1 design has the expected number of patients around 10 patients as compared to about 12 patients for the classical 3+3 design. So essentially it means that this 3+1+1 rule gives us a little bit smaller sample size with relatively with a similar performances in terms, for instance, of selection of MTD in one-third of the cases. These are the main. So these are the disadvantages of classical 3+3 design, and as I said, the design is very often used just because of its convenience and very easy implementation. Nevertheless, it's not very efficient, and there are better designs which are available there, and I would like to talk about two of them. The first one is the so-called continual reassessment method, which has been proposed in 1919, so already 20 years ago, more than 20 years ago, by John Quigley and colleagues. In the CRM, in the continual reassessment method, the dose given to the next patients, single patients or a cohort of patients, is based on an assumed dose effect curve for dose-limiting toxicities. So essentially, we assume some kind of a dose-toxicity relationship. And the idea is to assume some initial curve and to reassess the relationship in the light of the toxicity observed in successive patients, and therefore to guide our dose selection by the best current estimate of the toxicity of the doses. So you've seen already this picture. So essentially, we could say that we postulate kind of a dose relation, dose response, dose toxicity relationship, and we may want to assign doses which are close to our target percentile of the curve or probability of DLT. It may be 33%, it may be 20%, it depends on your choice. And essentially this curve, this dose toxicity curve, is some kind of a parametric curve which depends on a single parameter, and the idea is that by updating the value of the parameter when based on the observations which we make in the trial, we can actually better estimate the MTD. So essentially, before the trial starts, what we should do is we should make an informed guess based on the preclinical data, for instance, about the probability of DLT at each dose. So it may be that, for instance, if we start with 5 dose levels, we could just number these doses and say we can have some initial guesses for this probability of DLT. This will very much be useful only in terms of having like a scale on which we'll work with those toxicity curve. And in the second step, we choose a model to describe the relationship between the probability of the toxicity and the dose. This can be, for instance, this single parameter logistic regression, where we have logistic model where we have A as a parameter, and essentially where X is the recoded dose. So, for instance, it is that the dose may be recoded based on the prior probabilities, which are assumed for the dose toxicity curve with parameter A equal 1. So essentially what you say is, okay, these are my potential dose toxicity curves, and in the process of the data collection we'll be trying to update our model, so we'll be selecting these different curves, and based on the curves we'll be selecting the doses for the next patients. So essentially what then happens is that you choose an upper limit for the probability of DLT which you want to achieve. For instance, this can be 33% or 20% or 25%. You can choose the percentile you want. And essentially, the next cohort of patients is assigned to the dose for which the dose-limiting probability is closest to the target, right? Now, this cohort is observed to experience a DLT or not, and based on these data, and also using the data for the previous cohorts, the model is refitted, and the updated value of the parameter A is obtained, and then it is used to update the probabilities of the different doses, right? And we assign the dose for the next cohort of patients by looking at these updated probabilities of dose-limited toxicity and selecting the dose for which this updated probability is closest to the target, to the 33%. And then we repeat this procedure until some stopping criterion is met, and essentially at the end of the procedure we will have a dose for which the probability estimated probabilities closest to the target. Now, in the continual reassessment method, there is no inherent stopping rule. There are several possible criteria. Very often what is used is a fixed total sample size is taken. So essentially you say, okay, I want to put on in my trial 24 patients, and you essentially run your trial with 24 patients, and when you reach 24 patients, you, according to the updated model, you select your MTD. So you recommended dose for the phase 2 trial. There may be also a fixed maximum number of patients treated at a particular dose or at the MTD, so you may actually require that you want to have at least, I don't know, 8 patients treated at MTD. You may want, there are more advanced rules, like for instance a fixed precision of the DLT probability estimate may be sought for, so you might want to stop the trial when you achieved a particular precision on the DLT probability at your MTD. And also you can have a rule which says that if the probability of assigning the same dose level for the next, for instance, 4 cohorts, if this probability is high, like 95%, which means that you wouldn't really change your dose anymore, then you can stop. And this essentially means that you've reached your MTD. So any of these rules can be applied, and they differ only in let's say the ease of implementation and in the, let's say in the expected sample size that you might expect. Now, there are modifications of the basic CRM design. The CRM is a model base, as you've seen, and it's a very versatile method which can implement many practical constraints. For instance, you can start at a low dose. You could also start at the dose at which you a priori think you are closest to the MTD. The dose increases can be limited to one dose level. In principle, CRM allows you to skip the doses when increasing, but if you are worried about the possibility of jumping too high, you can do this just by limiting to one dose level increase. You can have accelerated titration for initial dose increases so that you can go quickly with respect to approaching the MTD level. You can have at least 2 patients or 3 patients per cohort to be assigned to a dose level. Originally, CRM was proposed with a single patient cohort, but this might take too much time, especially if you want to observe DLT within some extended period of time. So in order to speed up the trial, you might want to use cohorts of 3 patients, for instance. You can overrule the model recommendations, so it may be that, for instance, according to the model, a particular dose increase, the model can suggest increasing the dose, but it can be that it is possible to say, no, I still want to assign the same dose because maybe there is some doubt about the toxicity profile of the patients who were treated at this dose previously, and this is no problem. You can actually do that, overrule the model recommendation, and then run the next cohort with the same patients, and then after these patients are treated and observed, you can again update the model and see whether you would go for an increase or not. So there are many potential modifications which can be used, and essentially it has been investigated by extensive simulations that these modifications do not change the performance of the CRM design very much. Of course, some of them might increase, for instance, number of levels which are visited, number of dose levels which are visited during the trial, but essentially from efficacy point of view, there's not much loss. Now, what are the advantages over the 3+3 design? Well, compared to this design, CRM-based designs find the true MTD more accurately. And more importantly, they treat more patients at optimal dose levels, so essentially it means that you can offer more patients doses which may be close to a therapeutic dose, and therefore this idea of treating these patients even within phase 1 trial might be realized. You can reach the MTD in fewer patients, especially if MTD is at higher dose levels, because with the 3+3 design, you will have to visit all those levels before you reach this MTD level. Whereas in CRM design, you can actually get there quicker, especially if you allow, for instance, dose skipping. and that CRM-based designs are flexible, so you can adapt them to any target probability of DLT. They allow for modifications of dose escalation recommendations. All these modifications which I mentioned before are possible within this design, whereas for the 3+3 design, you essentially are stuck with this scheme of assign 3 patients and then add maybe more three more. This is illustration of this feature of the CRM design, that more patients are put on at the recommended dose level. So as you can see, you have this is a classical CRM with 1 patient assigned to the dose, and you see that we start, let's say, with the 4 dose levels. You would increase in the first 3 patients to the 3rd dose level, then to the 4th level, and if it appeared that the 4th patient would experience would experience DLT, the model might indicate that you need to go to one dose level down. Then you could stay at this level, and then if there's nothing happening, you would go up perhaps. Then you might see 2 extra toxicities. Then you would go probably down, and then you would very likely stay at this level. Well, in this case, you stayed at this level. So as you can see, there are many patients which are treated essentially at this level, which will be selected as MTD, and this means that essentially you will have more information about the toxicity at this MTD level than in the classical 3+3 design. Now, what are the disadvantages of CRM? Because I was sort of like praising CRM very much until now. Well, essentially, I can see only 2. One is the more advanced implementation, because you indeed need a software which would allow you to update the model. Although the software is not a specialized software, you can actually very easily implement the design in a classical using classical SAS procedures. And perhaps more frequent interaction between statisticians and clinicians is needed, because if statistician runs the software for model updating, then of course there has to be some coordination. Now, I don't see myself this as a disadvantage, because I can only recommend to you more interaction with the statistician when you organize a clinical trial. But indeed, this means that from the logistic point of view, this is an additional perhaps complication which needs to be taken into consideration when organizing a trial. So that might be perhaps a drawback. As I said, I don't see it as one. So CRM is a very nice statistics-based design which actually beats 3+3 on many grounds. Recently, and this is an example, very interesting example which has been published by Xavier Paoletti and colleagues. The target DLT level was 20%, and as you can see, the doses which were used in this trial range from 10 to 6,400. So there was a huge uncertainty about where the MTD might be. And essentially what happens is that what you can see is that initially single patients were put on the doses, and as you can see, there was very slow initial dose escalation, which means that they started with a too low initial dose, and it took them a lot of time to reach the first DLT level, and only then they started to put more patients at the levels which are around this area where the toxicity was appearing. So what we can see is that there was a huge dose increase from 10 to 4,000 before the first DLT was reached, and this is a situation in which you certainly wouldn't like to do 3+3 design. And MTD, which was dose 13, this dose of 5,600 units, it was reached after 37 patients. So the trial was stopped after 37 patients were included in the trial. And you can see that, well, if you would consider like 14 doses with at least 3 patients for a dose in a 3+3 design, you might expect over 40 patients in such trial. So in this case, essentially the CRM allowed to reach the decision quicker than the classical design would allow. This is, by the way, a very nice paper where you can see all the details of the implementation of the CRM in this particular trial. Now, recently, this design, modified toxicity probability interval, has been design has been proposed. It sort of like looks at improvement of the efficiency of the 3+3 design, but also tries to aim at the practical implementation issue. So essentially, what happens is that before the trial starts, the so-called equivalence interval is selected for the target DLT probability. So essentially, let's say that if you would aim at 30% DLT probability, this equivalence intervals might be something like 25%, 35%. So 30 minus 5%, 30 plus 5%. And essentially, the equivalence interval means that any dose with the probability of DLT within this interval can be selected as the MTD. So for clinicians, this is like a clinical equivalence interval where any dose is good for treating the patients. Now, for those adaptations in the trial, the following intervals are considered. So you have the equivalence interval, which is where the doses are close to the MTD. You have the interval for the probabilities from 0 to the lower end of the equivalence interval. So in our example, it would be from 0 to 25%. These would be the doses lower than the MTD. So you would not like to, you would like to escalate if you are in this region. And you have the complement of the upper limit of the equivalence interval. These are the doses higher than the MTD. So if you land in this interval, you would rather want to de-escalate the dose. So the whole idea of the trial is that after observing toxicity results for the last cohort of patients, you compute the probability mass for these three intervals. So this is a little bit complex maybe, but this is a picture which illustrates the situation. You have here these three intervals, so lower doses, MTD equivalent doses, and higher the MTD doses. And then you can calculate based on the actual likelihood of your DLT data. You can calculate the probability mass which falls on this interval, on the equivalence interval. It would be this probability, and on the higher than MTD doses interval. And then you essentially escalate dose if the probability mass is the largest for this lower the MTD interval. You stay at the dose if the probability mass is the largest for the equivalence interval, and you reduce the dose if the mass is the largest for this too high dose interval. Okay, so then you essentially recalculate the probability mass, and you assign next patients to the doses which are corresponding to which fall into the particular interval. And at the end of the trial, essentially what you do is you select as MTD the dose for which the difference between the probability of DLT for the dose and the target probability is smallest. And you do this for all those which you tried and for which this posterior probability that the probability of the DLT for the dose is greater than the target is small, like 5%. So you essentially look at the doses and you select the dose which fulfills these conditions and for which the probability that the probability of DLT is greater than the target is small. All right. Now, this may sound complicated, but it's essentially not very much, because what happens is that you can actually pre-compute your dose escalation rules. So this is an example of a table which you could pre-compute before the trial is run. So here is the number of patients treated at the current dose. Let's say the trial is aimed at 30 patients, sample size is 30. And here you have number of toxicity which are observed in a particular dose. So you can see that, for instance, if you had like 12 patients treated at the current dose, and out of them 0, 1, or 2 patients had DLT, you essentially would escalate, all right? On the other hand, if you would see 3, 4, or 5 DLTs out of 12 patients, you would stay at the same dose and you would treat the next patients at this dose. If you would have 5, 6 DLTs, you would have to de-escalate to the next lower dose, and you would have 7 or more DLTs. This would mean that you should de-escalate, but also you should remove this dose and higher doses from your trial, because this dose is, the current dose is unacceptably toxic. So this design also allows you to essentially decide at a particular moment to remove some doses from your trial as too toxic ones. So this table can actually be computed pre-computed before the trial is run, depending on the sample size and the target you are aiming at. And this can be actually provided as the implementation, so there's no need to run a program to update the doses during the trial, but well, to calculate these doses, this table of dose assignment, you do have to do some calculations beforehand. So there's no escape from collaboration with the statistician here as well. So what are the advantages over the 3+3 design for this modified probability toxicity interval? Well, again, compared to the 3+3 design, the MPTI design is based on a statistical principle, which is a very good feature anyway. It treats fewer patients at doses higher than the MTD. So it means that it protects the patients from overly toxic doses, and it yields a higher probability of identifying the true MTD. So essentially, it is more efficient in the sense that you will reach your conclusion in more percentage of the cases. What is the disadvantage? Well, again, you might say that this advantage is a more advanced implementation, because this pre-computation of the dose escalation strategy is possible, but you have to know how to do this. So essentially, someone with statistical background should help you. But as such, I don't see this very much as a problem. Now, what is the comparison of CRM? And this is actually a very interesting quotation I found in the paper investigating the properties of the MPTI design. So in this original paper, essentially what the authors say is that they believe that the CRM is an excellent method with many advantages over 3+3 like designs. So even in this paper, they indicate that CRM is actually better than the classical 3+3 design. And furthermore, our simulations show that in certain situations the CRM can outperform our method. So indeed, the MPTI design is actually is a good one. It's more efficient and in many ways better than 3+3. But as you can see from the simulations of the authors conducted by the authors, CRM can also be better than this modified toxicity probability interval design. So essentially, one could say, why not using CRM in the first place? So this is basically what I wanted to say. Our tentative conclusions, and this is where I'm coming back to the part which Everard also which Everard also presented. Well, phase 1 trials are, of course, a critical translation point of preclinical into clinical research. And obviously, an adequate phase 1 program is key to the successful development of a novel agent. You don't want to make a mistake and decide that the agent is too toxic, for instance. Statistics principle designs for finding the MTD are more efficient than the classical 3+3 design, but they are underused. And this is a sad conclusion which I could offer from my experience. There are designs which are much better than 3+3. They are slightly more complex to be implemented, but they are much more efficient in terms of finding the MTD more often and reaching the MTD quicker, so with a smaller number of patients. So for the price to be paid, just a little in terms of a little more complex implementation. But I do believe that this price is worth paying, and therefore collaboration with statisticians in implementation of these designs like CRM or modified probability toxicity interval is worth considering. That's all what we wanted to present to you. Thank you very much for your attention. Okay, I think, well, we were thinking about having some questions at the end, but I'm afraid we are over the time. So if we can suggest to you to send us your questions by email, or we will collect the questions from the chat, and we will try to send you answers by email. And of course, you can always contact us if you would like to have some support with the designing or planning of a particular phase 1 trial you are interested in. Thank you very much for joining us.